Okay, so let's get started. So first of all, we'll talk about Huntington's disease. A very important point for HD. This is called as HD, Huntington's disease. Okay, in this disease there will be important feature of chorea, Huntington's chorea. Okay, this is autosomal dominant inherited disease. I've already created a lecture about autosomal dominant. Hope you have watched it. If you have not, please go and watch it. Also, HD is a progressive neurodegenerative disease. Progressive means as the age progresses, the diseases will become much more severe. Okay, how we'll get to know that. Okay, next is neurodegeneration. In the neurodegeneration, so nerve get degenerated or destroyed, degenerated or destroyed. So all the manifestation of this disease is because of neurodegeneration. Okay, so all the disease manifestations will be because of this disease. And in the manifestation, because the nerve is involved and nerve is responsible for a lot of function. Okay, there can be physical impairment, physical, mental, psychological, etc. Okay, so because it is an autosomal dominant condition, so there has to be some sort of gene involved, right? So gene is HTT gene. Okay, HTT. Where it is found? It is found on chromosome number 4. So all these 2 points are very high yield. Okay, chromosome 4 HTT gene. Okay, fine. So we are done with this. Now, what actually happened in this HTT gene? Okay, we will get to know about this. So normally in HTT gene, so let's say it is an HTT gene, okay, and there is CAG repeats. So CAG repeats, and repeat means after one repeat, so this is called one repeat. Okay, we will have another CAG. Okay, so this is CAG and this will be a second repeat, and like this it will go on and go on. CAG again, CAG, up to how many times? Normally it is from 10 to 35. Okay, so normally it is 10 to 35 repeats. Good. As we have talked about the repeats, so I would like to throw some light on this topic that in the non-Mendelian type of disorders we talked about trinucleotide repeat disorders. So okay, so Huntington disease is one of them. So there are roughly about 4 different types. Huntington disease is one of them, and also other can be myotonic dystrophy. There is again Friedreich ataxia, then there is a fragile X syndrome. So when we say repeats, so in this case CAG repeats are much more prominent. Okay, so CAG repeats in case of Huntington disease. In the cases of myotonic dystrophy, it can be CTG. So CTG in case of myotonic dystrophy. Then for Friedreich's ataxia, there is a GAA repeats. And lastly, for Fragile X, it is CGG, Fragile X. We'll talk about them later, okay, in some other video in much more detail. So we were talking about the CAG repeats. So this was considered normal. So whenever there is some kind of disruption in the gene because of some sort of mutation, genetic acquired things, and so, these repeats increase in number. It can range from about 36 to 100 and even much more higher. Okay, this will become abnormal, and because of this, the protein formed by this gene will be affected. So let's say this CAG repeats time n times, which is roughly around 36 to 100, 36 to 100. This will code for a defective protein. Normally this protein will give up a normal protein, okay? This will be normal, which will cause some sort of, which will carry on some sort of functions in the neurons. In this case, which is the abnormal one, abnormal 36 to 100, CAG repeats, right? CAG repeats. It will make up a protein which is again abnormal, and this abnormal protein will keep on accumulating, within the neuronal cell. Let's say this is a neuronal cell and this is the defective protein. It will keep on increasing, and after some time the cell will die because of this toxic protein, okay? So cell will start to die, and this is called as neurodegeneration, okay? There are other types of neurodegeneration diseases also. We'll talk about them someday later. So in Huntington disease there was a neurodegeneration, right? So we're done with this one. Now what actually happened is this, that primarily it will affect major parts of important part of our brain. So important part over here will be striatum. Striatum will be composed of again 3 to 4 parts, main 3. So which is the caudate nucleus, then there will be putamen, nucleus accumbens, and this will be divided, okay? This is the divided by internal capsule. So we must know that all these 4 parts, I'll show you them in imaging things. Now where were we? So what's the function of striatum? Striatum is very important because it's a part of basal ganglia, right? And basal ganglia is a very important structure of the brain which causes the fine tuning of the movement. So basal ganglia will cause the fine tuning of movements. Now because the structural component of basal ganglia of the striatum is disrupted, right? So after it get disrupted, so caudate nucleus will not function properly, putamen will not function properly. So the fine movements, or also we can say the unnecessary movements, are actually inhibited. Unnecessary excitatory movements get inhibited, so that only required or desired movement that we want in our body can happen. So this is done by the striatum, right? So if these get destroyed, neurodegenerated, so the function will be accordingly, right? Now let's see some images regarding this. So let's see. So this is an image, right? So here is the image of basal ganglia. Let's mark some structures, okay? This whole part is your striatum, okay? Striatum. And within striatum there are several other parts. This part is the caudate nucleus. This part is again this is putamen, and the rest of the other part, this one, will be our nucleus accumbens. So usually this one is normal, okay? And this one is the Huntington's disease. Now what will happen is that these structures will finally get destroyed. Oh my god. So these structure will get destroyed, okay? And once it will happen, the structure which will have higher pressure zone, which is the lateral ventricle in this case, it will exert pressure peripherally, and because of this, the overall structure of the lateral ventricle will increase. Okay, so size will increase. So this was the increase in size of lateral ventricle. Now you know what is the cause of it, how the lateral ventricle get increase in size. Now also we know that the main is the striatum. However, as the disease progresses, the function of the brain will much more hampered. Now we just said that striatum inhibit unwanted movements by fine tuning the response coming from the cortex. Now if striatum is destroyed, okay, or atrophied, there will be uncontrollable dancing movements, okay, which is called as chorea. Now how it actually presents, let's see one more image. Now this is a very typical picture of dancing movements. So this child in over here cannot control his movements, okay, and cannot fine tune them. So he can seen in maintaining different types of bodily movement, okay. You can see the legs, hands, face features, and so and so. So this is uncontrolled movement, chorea. Fine. What else? Let's put another background color. Fine. Now one more point, we just said that there was a progression of the disease. How this happens? So progression depends on number of CAG repeats. A very, very important feature. More the number of repeats, more will be the severity. Okay, it is directly proportional. Now it is also responsible for early onset. More the number of repeats, early the onset. Early onset of disease, and this whole cycle will keep on going and going until and unless until the patient or the person who has the disease die before giving this autosomal dominant condition to the offspring. So in future there will be no offspring, and this sequence will come to the end, and finally again the person who will acquire somehow, or it get it from the other person who has the disease, it will be again carried on to the generation until unless the last offspring will also die before giving birth who has the disease. Fine. So we are done with this one also. Now it is very important that to know that at which repeats, how when will be the onset. So about 36 CAG repeats, a little bit more than that, the onset will be somewhere around 30 to 50 years of age. For more than 60 CAG repeats, the onset will be somewhere around, so there's more repeats, the onset will be much more early. So this is also known as juvenile Huntington disease, okay, and so. Now, what are the some clinical features of Huntington's disease? Let's see them. So we just said that clinical features, clinical feature. So first of all, there was a chorea, right? Why? Because of neurodegenerations of basal ganglia, striatum in particular, right? The caudate nucleus and so and so. So there was a chorea, then there was eventually motor areas will be also affected. Motor speech disorder will be there. And because of motor speech disorder, so the muscles of speech will also be hampered, and ultimately the area nearby will also get affected. Person will have aspiration, or we can say there will be chances of aspiration. So there, he can have pneumonia later, and it can also lead to death of the patient. Okay. Also there will be rigidity, right? Good. Then there will be depression, anxiety, memory loss. Also, as we just said, that there was a motor speech disorder, so there will be problem with swallowing, so dysphagia will be there. The list goes on like this. Okay, there will be more and more and more. So as disease progresses, the striatum part of the brain will get atrophied, striatum atrophy. However, this was in the early phase. In the late phase, whole brain will get there will be a global atrophy. So which will lead to shrinkage of neuronal or brain parenchyma. Fine. Now there are certain diseases and disorders which have the similar presentation in this case. I will just discuss that there was atrophy of caudate nucleus, right? In this case, what actually happens is that there are so many other types of differential diagnoses which we can make for the same sort of disorders. For example, let's change the color. Okay, let's switch with which one? Let's change it with green. Okay, green is better. So differential, oh my god, differential diagnosis. Differential diagnosis of Huntington disease. Okay, so there are many of them. First of all, Parkinson, then Alzheimer, then also Sydenham chorea. So there are two types of chorea, right? First one was Huntington, and second one is this one, Sydenham chorea. For reference, just know this is due to infection, streptococcal, post streptococcal infection, right? Similar symptoms can be also due to HIV associated chorea, associated chorea. It can be due to stroke as well. Then we talked about Parkinson, Alzheimer, so and so. There can be also some psychogenic movement disorders. Cool. So we are done with these one. So these are your basic differential diagnosis of yeah. And one more thing, we just said that there was increase in size of lateral ventricles, right? So it is, it can be very very easily, misinterpreted from hydrocephalus. So while making a diagnosis, we just see all the clinical key features to rule out other conditions. So how we diagnose it? So for diagnosing, we just have certain things. How to make a diagnosis. Now, for making a diagnosis, first of all, we just see clinical presentations, all the features we talked about. It is important to know that maybe some features may be present, may be absent, okay, but the chorea is a key feature, okay, it will be present. However, there may not be depression, there may not be anxiety. It totally depends upon the severity and the extension of the disease, okay. So clinical presentation, okay. Now because it was genetically acquired autosomal dominant disease, so family history will be positive in this case. So positive family history, fine. Also, it is important that what's the age of patient, right? When we are done with this one, we have a suspicion, okay, there can be something, right? So we check for other symptoms. So symptoms evaluation, how severe the symptoms are, and this has to be done along with the clinical presentation, okay. Then we can go with the CT and MRI to check for CNS pathologies, atrophy of regions, okay. And after all these things, still it is not clear, right? It can be other differential diagnosis. So however, because the family history is positive, there is certain clinical presentation, okay, we can now make a high suspicion of HD, and we can directly order genetic testing, okay, in which we can check for chromosome number 4 CAG repeats, how often the CAG repeats. If it is more than 36, we have a, so genetic testing is the gold standard over here, gold standard to make a final conclusion and diagnosis. So we're done with this one. Now finally, what's the treatment for this? As it is genetic, so no cure. Patient will eventually die, or even if he is alive somehow, the quality of life will be extremely hampered in this case. So no cure. However, we can give symptomatic management, and we can also give tetrabenazine to control the chorea, okay. For depression, we can give SSRIs, antidepressants, and for chewing problems, speaking problems, we can go with physical therapy and speech therapy, okay. These can somehow target to limit the disease, but again can't help the patient much. Okay, so this is all about Huntington's disease, right? Let's see, did I just forget to share some other important images? No, I guess it is done. So yeah, thank you so much for listening about autosomal dominant. Next we will talk about other disease. So we are done with Huntington's disease. We'll talk about neurofibromatosis type disease, which is again of two types, NF1 and NF2. We'll talk about enough in much detail. Okay, so thank you so much for listening. See you in the next video.